But remember, the whole point of all of this is that we need to make our eventually 30 ATPs. And remember, we in glycolysis and in the Krebs, we managed to create a total through substrate level phosphorylation of 4 ATPs. So we need to make 26 more. So we need to now calculate how are we going to get those ATPs. Before I jump into that, I just wanted to give you the names of the 4 big complexes in the electron transfer chain. I already gave you the names of the 2 mobile carriers, but I put the names of the 4 big complexes on the board so that you'd have these in your notes. Complex 1 is called NADH coenzyme Q reductase. Complex 2 is called succinate coenzyme Q reductase. Complex 3 is coenzyme Q cytochrome C reductase. And then complex 4 is cytochrome C oxidase. So have these in your notes. There are lots of other smaller things that are going on within each complex. You can read about all of that in your book, but these names you should know associated with the complexes. Okay, now remember what happened in glycolysis. Net in glycolysis, I have a summary over here. If we started off, remember how we started off with 1 glucose molecule? All right, we said that that glucose molecule got turned into 2 pyruvate. So going through the 10 enzymatic reactions of glycolysis, what was the net product aside from the 2 pyruvates? We got net 2 NADHs and net 2 ATPs. So that's what we got out of glycolysis. The transition reaction, that pyruvate dehydrogenase, remember fate of pyruvate in an aerobic muscle cell. Pyruvate dehydrogenase took the pyruvate and turned it into the acetyl-CoA. Remember, 1 NADH was produced, but remember how we had 2 pyruvates, so we had to go through that twice. So in the transition reaction, you produced 1 NADH, but you did it twice, so a net total of 2 NADHs. In the citric acid cycle, Krebs cycle, what did we do? Well, we have to go through it twice. Why? Because we have 2 acetyl-CoAs coming from that 1 original glucose. All right. In one loop of the citric acid cycle, you only got 3 NADHs, 1 FADH2, and 1 ATP. But since you had to do the cycle twice from the 1 original glucose that produced 2 pyruvates, 2 acetyl-CoAs, each acetyl-CoA, remember, goes through the cycle once. So total 6 NADHs, 2 FADH2s, and 2 ATPs. Remember, the ATP came from the GTP. Okay, now, how are we going to get the other 26 ATPs? We've already got these 2 and these 2. Well, this is the deal. When those protons are going through that F0, F1 protein and allowing that energy to be captured by the ATP synthase to help put the phosphate group onto ADP, this is how we have to crunch the numbers. For every NADH that's produced in the mitochondria, so the NADHs that are produced in the transition, NADH is produced in the citric acid cycle. So we've got 8 NADHs. For every NADH in the mitochondria, there are 8 of them. You see that 2 plus 6 is 8. We're going to multiply this by 2.5, and that will give you the number of ATP you're going to get. So for every 1 NADH, you're going to get 2.5 ATPs. For every 1 mole of NADH produced in the mitochondria, you're going to get 2.5 moles of ATP. So do the math. 8 times 5, 40. Okay. There we've got 20 ATPs. Everybody see that? Okay. Now, for every FADH2 that's produced, you multiply it by 1.5. Okay, So it looks like we've got two FADH2s here, but we forgot about these guys. Now these NADHs, remember they were produced in the cytoplasm. In order for them to get into the matrix of the mitochondria, several steps, and they become FADH2s. So let's add them up. So 2 plus 2, that's 4 of them. That's 4. Okay. 4 times 1.5 is 6. Okay, one more time. The NADHs that are produced in the cytoplasm, in order for them to get into the mitochondria and then eventually get, to the electron transport chain, several steps, we count them as FADH2s. Okay, so this is counted as an FADH2. These are 2 over here. 2 plus 2 is 4. Multiply it by 1.5, you get 6. So that's a total of 26 ATP. We had these 4 that were produced through substrate level phosphorylation. These were produced through oxidative phosphorylation. These 4 were produced through substrate level. We add the 4, and we have a total of 30 ATP, which is what I promised you at the beginning when we first started carbohydrate metabolism back in the previous chapter. Okay, great. So that means for every 1 glucose, you're going to get 30 ATPs. What if you had 10 glucoses? 10 times 30, 300 ATPs. What if you had 100 glucoses? That would be 3,000 ATPs. Did you get the idea here? All right, good. So that covers what you're going to need to know about how to get ATP out of carbohydrates. In the next chapters, we talk about how we're going to get some ATP out of all of the lipids and the proteins. But right now, I just wanted to focus on all the carbohydrates. So we tied that into the chapter that we had covered in the last set of videos in the carbohydrate metabolism set. One other thing I wanted to tell you is that the citric acid cycle, if you just do one cycle, and I know I'm repeating myself, but I just want you to know that one cycle, so from one acetyl-CoA, you're getting 3 NADHs, 1 FADH2, and 1 ATP. This is from one acetyl-CoA. Because on your list of what you need to know, it says, what does it say here? It says here, be able to calculate the number of ATP molecules produced from different molecules. So if I say, all right, how many ATP molecules could you get from one acetyl-CoA? Or how many ATP molecules could you get from one pyruvate? Or how many could you get from one glucose? You know how to go through the system here. One acetyl-CoA, all you would have to do, remember these are produced in the Krebs cycle, so these are all in the mitochondria. You multiply this by 2.5, you multiply this by 1.5, and then you just add this, and you end up with 10 ATPs from one cycle. Okay, just double check my math here. Yes, you'll get 10. Okay. Last thing on the what you need to know list says, or actually the next to last, or I jumped over it, the uses of ATP. What is the whole point of having ATP? Let's write down some key functions of ATP in your body. Number 1, movement. You cannot, again, from your A and P classes, actin and myosin, when they slide against each other, you have to have ATP there, so you need it for movement. Okay, those of you who are constantly moving, are fidgeting, you're using lots and lots of ATP. If you're a couch potato, you're not moving a lot, you're not using a lot of ATP. So get up and move, you'll be using a lot of ATP. Okay, number 2. You may have heard of this in a bio class, but you need ATP for active transport. So if you want to push something against a concentration gradient, you need an ATP to allow things to enter cells or enter certain areas against a concentration gradient, and that's called active transport. You need ATP for that. Number 3, anabolism. Remember how when we first started talking about metabolism, we said metabolism is catabolism and anabolism? Catabolic reactions are basically what we've been doing so far. You ate a meal filled with carbohydrate, you got all that glucose, you then did catabolic reactions, you broke it down, and you ended up with energy. So catabolic reactions allow you to have an output of energy. Anabolic reactions need an input of energy. So in order to synthesize larger molecules in your body, in order to put small molecules and make bigger molecules, in order to synthesize many, many things, you need to put in ATP in order to do that. So all those anabolic reactions need an input of ATP. So these are 3 of the most important functions of ATP in the body. There are others, but those are the key ones I want you guys to know. Okay, we are all set with this section.